So we're coming back to this idea of PI, isoelectric point. Okay. Now, isoelectric point, again, remember it's the pH in which the net charge of a molecule is 0. Okay. That does not mean the molecule is not ionized. Okay. So the molecule could be ionized. It's just that it has both negative charge and positive charge, so that the overall net charge on the molecule is 0. Okay? And PI immediately has a few things that is worth our attention. Number one is PI affects protein solubility at a particular pH. Okay, so essentially, if you have a solution that has relatively high pH or relatively low pH for the PI, okay, compared to the PI, then if it's high pH, the pH is higher than the PI, then your molecule will be negatively charged. Okay? If the pH is lower than the PI, then your molecule is going to be positively charged. Now, the same idea applies to a protein. Okay? Protein would have many, many charges on them. Right? And let's take a look at figure A on the top left. You see that when your protein is in a high pH environment, the protein will be negatively charged. So this protein is negatively charged. This protein is negatively charged, so is this. So they will all be negatively charged, and negative charges repel each other, right? So they repel. So that will keep the proteins away from each other, therefore increasing the solubility. Okay, so the proteins are more likely to be by itself inside the solution. Okay, whereas if you are at the isoelectric point, okay, if the pH is equal to the PI of the protein, then the protein is no net charge, but it still has ions on them, okay? Meaning it still has negative, positive, and your neighboring protein will also have negative and positive, and then they're going to interact with your neighbor proteins, okay? And they're going to form pretty stable complexes and lower their overall solubility. Your protein has no net charge, so they're just like floating, and then they have ionizable groups, okay, that are charged to interact with the other proteins. For example, positive on this guy interacting with the negative on this guy, positive, negative, right? Positive, negative. So they're gonna come together, and if proteins come together, the solubility drops, okay, and that would be bad for your chemical reaction, for example. Okay, so PI is important for protein solubility. So if you want to purify your protein or use your protein to do an enzymatic reaction, you probably want to pick a pH that's not exactly at the PI. Also, charges. Okay, we're going to talk a little bit about charges now. So charges on biomacromolecule happens all the time. Okay, biomacromolecules meaning it's a big molecule found in biology, right? If it's found in biology, you can imagine that it's probably very soluble. If it's soluble, a lot of times it would have charges. For example, your DNA. DNA is entirely negative charged. Okay, so it has no positive charges at all. Okay, so it's all negative, right? And because DNAs are negatively charged, so essentially if they're in a solution, they're going to repel each other. Okay, so DNA is actually quite soluble in water. Okay, so free DNA are negatively charged, thus repelling each other. If you have a lot of basic residues, okay, that are going to be charged at physiological pH, okay, and they're going to be positively charged. So what they can do is they have positive and they can attract your DNA, okay. So these proteins can then bind your DNA, stabilize your DNA, right? These DNA binding proteins have many basic residues, and you should know what the basic residues are, okay, like amino groups, right? Which makes them positively charged at physiological pH. This attraction helps their association with DNA, and you have heard about histone proteins, right? So histone proteins a lot of times have quite many positive charges on them so that they can interact with DNA, right? So this is something that we actually see a lot. These large polyelectrolytes, okay, such as nucleic acids, or polyenpholites, such as proteins. Now, notice the difference between the term, okay? Nucleic acids are polyelectrolytes, whereas the proteins are polyenpholites. Okay, remember, enpholites are the type of molecules that can have both acidic and basic residues, right? But DNA doesn't have that as much, okay? So it's primarily negatively charged, so we just call it the polyelectrolyte. So it's an electrolyte, right? Whereas proteins, it's polyelepholite, okay? And they're classed together as macro ions. So like macro meaning big, and ions, you guys know what ions are. So they're like big ions. Each macro ion collect about it a counter ion atmosphere enriched in the opposite charged small ion. Okay, so in a way, it's actually very intuitive. Let's say you have a huge DNA right here, right? This red thing's DNA. Let's say this guy's DNA. And you know DNA has a lot of negative charge around it. So essentially, it's going to attract a lot of positive charged ions, okay? And these could be small ions. Can you guys give me an example of a small positive ion? Okay, hydrogen, maybe. Okay, H3O plus, sure. Anything else? Negative small ions? You have chloride minus, positive small ions, Na plus, sodium ions, magnesium ions, calcium ions, right? So the metal ions are all positive ions, and then they're going to be small, right? Relatively to a DNA, right? DNA is huge. So metal ions are small. So then essentially what happens is you're going to grab a bunch of these little positive ions, sort of surround this entire cloud, okay? This entire blob of DNA, okay? And this cloud of ion tends to screen the molecule from one another. You rarely find like DNA just floating in water by itself, right? It's usually be surrounded by some kind of a counter small ion species, okay? And together they form this counter ion atmosphere, essentially. This is a term that you guys should know, ionic strength, okay? Ionic strength is a measure of ion concentration in a solution, okay? So why is this important? Because obviously a lot of our biomolecules are charged, right? They're either negative charged or positive charged. Anything that has a charge is going to behave differently in, for example, pure water or in an environment where you have a lot of other electrolytes, right? Because if you have other electrolytes, then this thing with the charge can be stabilized, whereas in pure water, it has probably more energy, right? So ionic strength will have something to do with the stability of your molecule. Doesn't have to be macromolecules, just any molecules in general. Ionic strength will have an effect on chemical reactions. Okay, so definitely they have an effect on chemical reactions. Now, so inside of a regular cell, typically the ionic strength, it's about 100 millimolar, okay, or 0.1 molar. So something like that. Okay, so that's sort of like a rough estimate of the salt concentration inside of your cell. Okay, so that's the physiological condition. Okay. All right, so it's a measure of ion concentrations, but like a lot of times it might not be easy for you to know exactly what ions are there because you have many different types of positive ions. But collectively speaking, we say it's the ionic strength. And of course, magnesium is 2 plus, sodium is 1 plus. That's different as well, okay? But just for your information, ionic strength just tells you about how much ions are in your solution. So it's like a little measure. So higher the ionic strength, obviously you will have a different behavior from a low ionic strength environment. That's ionic strength, right? So that's something you will run into in the future, okay, when you do research and stuff like that. So the stuff that we're actually going to talk a little bit about is electrophoresis. So electrophoresis, it's simply a way to separate chemicals based on their charge and size, okay? Now, electrophoresis is when you have an electric field, charged molecules will move. Well, I mean, duh, right? Your charged molecule is charged, so it's going to move away from the side that has the same charge and then move towards the opposite charge. Okay? So charged molecules will move due to repulsion and attraction. Negatively charged molecule move towards the anode, the plus end. Positively charged molecule move towards the cathode, the negative end. All right? So you can apply electricity sort of. You have an anode. Usually in an electrophoresis kit, it's a very thin wire at the bottom. And then you have a cathode, which is another very thin wire at the bottom. Okay? And then you apply voltage on it. Okay? If you have voltage, you have a separation of charge, and then you have an electric field. Okay? Now, if you have an electric field and you're going to throw something that is charged, for example, DNA, okay, then it's going to move. Okay, right? DNA is charged. Right? Now, DNA is going to move towards the anode or the cathode. Yeah, anode, okay? Right? DNA is negative charge, right? And this guy is like positive charge. So if you have a negative, it's going to go this way, okay? I mean, that should be quite simple, okay? This general process, we call it electrophoresis, okay? Using electricity to do stuff, right? So now we know that it works, okay? So yeah, things move, that's great, but then what do you do with it, okay? So it turns out the number one practice in a wet lab doing molecular biology or biochemistry is this thing called the gel electrophoresis. So we're going to throw your DNA inside of a gel matrix, okay? And this gel matrix can be made from different material. For example, if you're separating proteins, we tend to use polyacrylamide. If you are separating DNA, we tend to use agarose, okay, just different matrix materials that we use to separate DNA and proteins. And these gels, essentially they form porous matrix. So like a little solid looking thing that has a lot of holes inside, okay? That has a lot of holes. And a lot of holes allows your material to move around this medium. As electric current is applied over the gel, charged molecules, for example, DNA and proteins, in the gel will move through the gel, okay? And generally speaking, smaller molecules move faster, okay? Smaller molecules move faster, okay? Definitely. Why? Because you got all these holes, right? Small molecules can navigate around the holes very quick, whereas large molecules get stuck because the hole is only so big, and then your large molecule is like, oh crap, I can't go through, right? So it gets stuck. So large molecule eventually moves slower, but still moves. So this is gel electrophoresis. DNA is naturally negatively charged. When you're doing a PCR, for example, you want to see whether or not your PCR has worked. You're going to throw your PCR product onto a gel electrophoresis agarose gel, and then you're going to apply the voltage, and you're going to see your DNA is going to move, okay? That is because DNA is already negatively charged, okay? So you don't have to do anything to the DNA. Just throw the DNA into the agarose gel, DNA is going to move. Don't have to do anything. Proteins, on the other hand, is very different, okay? Proteins can be positive or negative charged, okay, depending on the pH, right? Of course, if the pH is like 14, then it's going to be negative charge all the time, okay? But proteins in general can be positive or negative at a specific pH. So you can't just throw protein on it and then say, hey, why can't you move? So it turns out you have to use a surfactant called the SDS, sodium dodecyl sulfate, SDS. You actually saw the structure of SDS when we talked about hydrophobic, hydrophilic. Okay, remember? It's a 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. Attached to it, dodecyl sulfate, and then a sodium salt. This thing, okay, can bind to proteins. Why is that? Proteins generally looks like a big blob, This will come back and visit when we talk about proteins, okay? But for now, I want you to know this. Proteins, if it's soluble, usually you have a lot of hydrophobic residues inside. Outside, of course, it's going to be hydrophilic. So if you want this protein to be charged, okay, that's actually what we want to do. We want to make sure proteins can run in electrophoresis, okay? So we're going to have to make our protein always either positive or negative, okay? Always, regardless of the pH. So we're going to use this molecule, SDS. SDS is always negatively charged because you have a sulfate here, okay? Sulfate always loses that particular proton, and it's always negatively charged at neutral pH. The reason why we use SDS is because you have a 12 carbon chain right here, like a fatty acid thing going on, right? So this guy is hydrophobic. Inside of the protein is hydrophobic, okay? So your proteins are going to be hydrophobic as well, at least some parts of it. Cool? We'll agree with that? So far so good, eh? So then naturally you put SDS with protein, and then usually we're going to boil it a little bit. We're going to denature the protein so that you expose the hydrophobic part of the protein with SDS lipid arm, okay? Such that what you get is something that looks like this, okay? So you're going to denature your protein. Actually, SDS is a surfactant, okay? Surfactants usually denature your protein as well. All right, so you're going to destroy the secondary structure and the tertiary structure of your protein, but you don't care because you want to separate, okay? You want to see your protein on a gel, okay? And gel electrophoresis does separation based on size, okay? Remember that? We said small molecules move faster, okay? Large molecules move slower because the gel matrix has pores, okay? So gel electrophoresis is a separation based on size. So then proteins, I don't care if it's still in the native secondary and tertiary structure. Doesn't matter because a folded protein of 300 amino acids and an unfolded protein of 300 amino acids, it's still 300 amino acids, okay? So we're going to denature it, and this protein is going to have a lot of negative charge on it. We throw it onto a gel, apply voltage, this thing is going to move, okay? Just like DNA would. Okay? So essentially you convert something that could be positive, negative charged, depending on the pH. I'm always going to make it negatively charged by using a surfactant. Okay? And of course, DNA, they don't just shine colors, right? You have to add a fluorescent agent to bind to your DNA so that you can see it. But as you can see, initially I put my DNA on the top, okay? I add DNA on the top, and then I'm going to apply my electric field, my voltage like this, and then DNA is going to travel, okay? And smaller DNA obviously travel faster, okay? So this is a result of a PCR, okay? So this top picture is what you get from a PCR, okay? So after you do a PCR, you load your DNA onto an agarose gel, this is what you'll see. On the bottom here, that's a protein gel. It's an SDS page. This is SDS page, which stands for polyacrylamide gel electrophoresis. Okay, so PAGE, page. Okay, so this is something that I did where I was doing a very simple protein purification, okay? So I have cell, okay? So this is total cell. For example, I only look at this guy, and this guy, okay? I'm gonna draw it on it's easier for you to see, okay? So let's say I only look at this, okay? So actually when I was doing it, this is just the cell. So it has all proteins. This is the soluble lysate. So I lyse the cell, break the cell open, spin down everything that's not soluble, and then everything that is soluble is right here. Immediately, what you see is you have a band right here that's not in the soluble. Okay, so that tells you that this particular band right here is insoluble. Okay, it's found in your cell, the original cell, but it's not found in the soluble protein lysate. So it must mean it's not soluble. Okay, simple. And then later, I don't know if we'll talk about it, but this is a technique that we call a polyhistidine tag purification. So I purify my protein, and this is the purified protein that I wanted. Okay, so all of this we can visualize using SDS page. So SDS page is a very good way for us to know whether or not your protein is expressed, whether or not your protein is purified, that kind of stuff. It's like one of the most routine thing you can possibly do in the lab. Sometimes, if your lab is quite rich, you're going to get this instrument called the capillary electrophoresis. So you guys know what capillary is, right? Capillary is like a little thin tube, like very, very thin. Okay, so it's just like electrophoresis. However, you don't use a big gel. Okay, you pack the gel or whatever separation matrix inside of this little capillary. Okay, so what you can do is you can do a very high resolution separation because your capillary can be very long. So previously here, gels, if you guys have been in a bio lab or molecular biology lab, you know gels usually like this big. It's not very big. Okay, the height probably 20 centimeters at most. Okay, so yeah, 20 centimeters, and when you apply voltage on it, okay, that material, it's going to be a resistor as well. So it has resistance, so it's going to heat up. As it heats up, the gel will actually break apart. Gels are not meant for you to get a very highly resolved picture. Okay? However, if you do it using electrophoresis using a capillary, you can actually do a very good separation because the amount of material you don't have as much. Okay? And the way it works is very simple. Okay? It's just like your gel electrophoresis where you have buffer on both ends and again anode, cathode. Okay? So depending on what you're putting, for example, if it's protein or DNA, it's going to go towards the end that it wants to go. Okay? And then you have a high voltage power supply, whatever. Okay? So what you're going to do is you're going to throw your sample into the capillary, and let's say your sample is positive charged, then it's going to get repelled by the anode. It's going to travel over there. As it travels over there, you have some kind of detector. Okay? This detector can be whatever, okay? As long as it can detect whatever that you are trying to elute. A lot of times it's going to be UV visible spectrophotometer, okay? Because proteins absorb light. A lot of molecules absorb light. Okay, absorb UV light especially. So you can hook it up to a UV spectral photometer. As soon as something comes out of the capillary, you get a signal. Okay, you get an absorbance. So you can record as a function of time what comes out. Okay, so that is used as an analytical instrument. Okay, it's an analytical technique. Right, so you can use this to sort of analyze with high resolution of what kind of stuff is in your sample. Right now, all we're talking about are different equipments or different types of things that you use to analyze things. The next thing we're going to talk about is called this isoelectric focusing. Right? Actually, this is the reason why I was talking about PI. Okay, so this is the reason. Since proteins have charges, okay, unless pH is equal to PI, therefore, they will respond to electric field depending on whether at this particular pH it's positive charged or negative charged. Okay? So isoelectric focusing will use a gel that contains a stable pH gradient. Okay? And you must be like, what are you talking about? Okay? You can actually buy this from a commercial vendor. Okay? So it's going to be like this. Like this. It's like a piece of gel, and the pH is different. Okay, so let's say you have low pH and high pH. So this gel has different pH. Okay, so that's the difference. In our previous examples of the gel electrophoresis, it's all the same pH. Okay, so like protein gels, DNA gels, okay, the stuff that we talked about 5 minutes ago, they're all 1 pH. Okay, now isoelectric focusing makes a gel that have different pH. How do they do that? You can go ask their company. Okay, maybe it's like a trade secret little thing going on, but essentially you just gel it at different pHs. But it's a gel that has a pH gradient, okay, low to high. So then you throw a sample. Let's say you want to do separation. You have a bottle of protein mixture, okay, right? And you have 4 different proteins. You want to separate them. Number one way is, of course, you can use gel electrophoresis, separate based on size, okay? But what if it so happens that out of the 4 proteins, 2 of them have the same size? So then you can use isoelectric focusing. You throw the mixture into the gel, apply the voltage, the proteins will start to move depending on whether it's a positive charge or a negative charge at that particular pH. So it's going to move. We say, okay, on my gel, this portion pH is 7, this portion is 8, this portion is 6. I have 3 proteins, circle, square, triangle. Pi is equal to 7. This guy, Pi is equal to 6. This guy, Pi is equal to 8. Okay? So I throw my 3 proteins at 7. Okay, I just throw it there. So who's going to move and who will not move? Okay? The circle protein will not move because it's already at Pi. So it's not moving. The square protein, is it going to be positive charged or negative charged? pH is higher than the Pi for the square, right? That means that the protein is going to get deprotonated. If it's deprotonated, it's more likely to become negative, okay? So this guy is going to get negatively charged. This guy is going to get positively charged. And I'm going to throw my electric field like this. The anode on the left side with the low pH, the cathode on the right side with the high pH. Negative, the square is going to move this way. Triangle, because positive, repel from the left, so it's going to go that way. Until the square reaches pH 6, it becomes neutral, stops moving. Triangle, pH equals to 8 equals to Pi, becomes neutral, stops moving. Okay? You effectively separated 3 proteins. Okay? So you can get 1 band, 2 band, 3 band, 4 bands. Okay, or in this case, 1 protein, the circle, 2 protein, the triangle, 3 proteins. So this is a separation based on Pi. Okay, so a very important thing in chemistry is that as long as your molecule have different characteristics, okay, you can use that characteristic to do separation. Okay, let's go over some examples. Okay, chromatography, you separate based on what? Liquid chromatography. For those of you that took organic chem. Usually, when we say liquid chromatography, you're separating based on like polarity maybe. Okay, of course it depends on what your solvent is. Okay, usually polarity. Okay, you separate based on polarity. Think TLC. That's why you separate based on. Okay, gel electrophoresis, you separate based on size. Okay, isoelectric focusing, you separate based on Pi. Okay, what about doing a distillation? You're separating based on boiling point, right? So as long as your compound or compounds have different characteristics, you can separate based on that characteristics. Okay, so that's very important. Okay, so that's the key about chemistry. pH of the gel, it drops, and this is the position of the gel. Okay, you can see how we can get pretty decent separation based on pi. 1 protein, 2 protein, 3 protein, 4 protein, 5 protein, 6 proteins. So you can separate them based on pi. I hope you guys understand that nowadays a lot of things are involving high throughput or large data analysis. Talk about this a little bit. Omics. Omics essentially is talking about the study that looks at a particular thing in its entirety. Okay, you look at this thing, everything. Okay, so omics, we have genomics. Okay, what does genomics look at? DNA, right? Okay, genomics looks at DNA sequence. Okay, so this is the reason why people were so interested in like the human genome, that kind of stuff, right? So we want to take an organism, sequence its genome, and then we can get, okay, it has this gene, has that gene, and then maybe we can get some interaction or information out of that. Okay, genomics. What do you think happens after genomics? Okay, so this guy deal with DNA sequence. Right? We have transcriptomics. This is looking at transcription. Essentially, it's going to look at your mRNAs. Okay? It's going to essentially look at expression of genes. Just because you have a gene in the genomics, okay, just because you have it, does not mean you're going to express it. Okay? This is transcriptomics. You have proteomics. This is going to look at proteins. This is looking at a cell. I want to look at what kind of proteins are there. Proteomics. Okay? I want to study all proteins. Okay? Proteomics, after proteomics, do you still have other stuff? Yes, we do. We have metabolomics. This thing looks at your metabolites. For example, your fat level, sugar level, your lactate level, your whatever. Okay, so all the chemical compounds inside your body, we can analyze it. That's called metabolomics. Okay? And there are actually a lot of other omics, for example, fluxomics, that studies how fast a reaction go in your body or how fast reactions go in your body. Okay? So omic studies are very important. And this is just a very brief, get you to see these terms, but we're not going to cover this, just so you know. Okay? Now, proteomics in particular, I want to talk a little bit about. It's the large-scale study of protein characteristics and functions. Under proteomics, you have this thing called the protein profiling. And protein profiling is frequently used to determine gene expression differences between cells, grown at different conditions, between different tissues, between normal and diseased or mutated cells. So we want to look at protein expression differences between two conditions. Okay, this condition could be normal versus sick, high temperature versus low temperature, high pH versus low pH, whatever. Okay, whatever you want to study. Okay, this is called protein profiling. Right now, 1D, one-dimensional gel electrophoresis is insufficient for providing the resolution necessary to see the different proteins expressed in a single cell sample or in a cell sample. Okay. Because your electrophoresis separates only based on size, okay? And it's very likely you have multiple proteins that have the same size, but they're different proteins, okay? So if you only do gel electrophoresis, you will not see enough information between normal cell and a sick cell. Therefore, we want to do a two-dimensional gel electrophoresis. Okay, and this is actually very frequently used. Right? This is very frequently used for those people that are doing comparative cell studies. Okay, and 2D electrophoresis combines isoelectric focusing which is separation based on pi, and SDS page, which is separation based on size. You first get your protein sample, and you're going to throw it onto isoelectric focusing, and you achieve separation by pi. Okay? So you have a separation by pi. And then you can separate based on different pi values. Right? And this is, of course, a gel. Right? This is a gel. And then you take this gel, you slap it onto an SDS page gel. On the x-axis, you have different pi. On the y-axis, this is going to be separation based on size. So now you have a two-dimensional separation. Okay? And this is a lot more powerful than a one-dimensional separation. So then you can see a gel that looks like this. And yeah, you're going to be like, I can't interpret this. Okay? But of course, there are softwares that can help you identify, oh, okay, maybe this blob is not found in the normal cell, but found in the sick cell. Okay? Then you know there's a difference between normal cell, sick cell. And the difference is this protein. Then what you can do as a scientist then is, okay, I take out this protein, purify it, and I find out who that protein is. And with our modern technology, we can get so once I get this protein, okay, I can get, using amino acid sequencing, we'll talk about that in the future, sequence the amino acid a little bit, and then I can find the gene that encodes this protein. And then I study, okay, maybe this gene has something to do with the normal versus sick cell. So using this logic, you can do that to all sorts of different studies. Okay, for example, in my field of work of biofuel development, I would be interested in, let's say I have a cell that does not produce biofuel, a cell that does produce biofuel, and I look at what's the difference. And maybe the difference will tell me something about, okay, if you have this enzyme, it produces biofuel. Then engineers can use this information to do engineering.